Ok, bom dia a todos. Eu vou fazer essa introdução em inglês, particularmente porque nós temos um convidado internacional, mas também porque acho que todos entendem bem o inglês. So it's my distinct pleasure to introduce today's speaker for the scientific session of Instituto Gonçalo Muniz, but also for the molecular epidemiology course, Dr. Wondwossen Gebreyes, a distinguished leader in the field of global health, veterinary medicine, and the One Health movement, a leader in this field of One Health whose work has really shaped the way we understand and respond to the interconnections between human, animal, and environmental health. So Dr. Gebreyes is the Hazel C. Youngberg Distinguished Professor at the College of Veterinary Medicine at The Ohio State University, and the founding executive director of the One Global Health Institute at this institution. He is a veterinarian and epidemiologist who is originally from Ethiopia, was trained at Addis Ababa University, and now leads international capacity building programs that are strengthening disease surveillance, antimicrobial resistance monitoring, and zoonotic disease prevention in more than a dozen countries throughout the world, including here in Brazil, in Paraíba, in Pernambuco, Rio Grande do Sul, many different states within Brazil. And it's our real pleasure to have him here in Bahia. His work really exemplifies the core principles of the One Health approach, from tackling drug-resistant pathogens and foodborne diseases to addressing the health impacts of climate change and biodiversity loss. Dr. Gebreyes has shown how transdisciplinary science at Ohio State University, but more broadly throughout the world, can be translated into action. His research has advanced our scientific understanding, and he'll present this during his lecture today, but also really has made concrete impacts on the health systems in resource-limited situations in Africa, in Asia, and here in the Americas. So Dr. Gebreyes serves in key advisory roles, such as the Forum of Microbial Threats at the National Academy of Science, and that's how I first met Dr. Gebreyes, and has received numerous awards in recognition of his contribution, including election to the National Academy of Medicine in the United States in 2021. And let me just end with just one connection, a personal connection. Wondwossen, or Dr. Gebreyes, is really, this is kind of our globalized world, but was very much a colleague of Lee Riley for the last 10 years, working together in advancing, expanding the work of molecular epidemiology in this notion, in this idea of One Health. And really, when I met, I think, Dr. Gebreyes last year, I was just surprised with how Lee, who was Lee Riley, who is someone very dear to our hearts, has been working closely with Dr. Gebreyes over the last decade before he passed away. So with that, it's a great pleasure to introduce Dr. Gebreyes. Thank you very much for making the trip to the United States to talk to all of us.
Muito obrigado, Albert, and bom dia a todos. Okay. Estou feliz, very happy for co-invite with Fiocruz. Great to be here. I wish to speak more Portuguese, and I wish this lecture was in Portuguese, but greatly appreciate. Very nice to be here. Nice to be in Salvador at Fiocruz. I heard a lot for so many years, and one key person was Lee Riley. At 1 year, he came, after this course, he came to Porto de Galinhas, where we had people from Brazil, other Latin America, from Africa. We gave a molecular epidemiology course. That was 2013. But over so many years, I've been really great working in Brazil with different states. Paraíba, where we started, we still do work in various parts of Paraíba, in the semi-arid, in the Caatinga biome, in various One Health-related parts, but also different states, Pernambuco, in Petrolina, where Bahia, even students are in Petrolina area, and further south, in Santa Catarina, Porto Alegre, and others. So Always very happy, very nice to be here. It's great to be connected, even though he passed through Lee Riley. And thank you, Albert. And last year at the National Academy, says you right away, we kind of in 5 minutes, we started discussing about this course and participation. So thank you for invite. Okay. So today I will be focusing on a little bit, of course, the focus is molecular epidemiology, but the role of global One Health, this interconnected planet we live in. We'll do a little bit of introduction and then give some examples of One Health related molecular epidemiology applications in some of especially bacterial pathogens. This article, a very detailed review article, it was supposed to be a book chapter. I'm sure everybody at PDF access, Luciano is sharing it. That was a book to be edited by Lee and Ron. So we ended up publishing it as a series. So it's several pages of a review article. So Lee asked me, you focus on zoonotic and livestock diseases because of my molecular epidemiology background. Okay. Let me start by, this week has been great listening to different topics, different molecular epidemiologic, observational, and surveillance and research. Wonderful talks in Brazil. Like yesterday, Fiocruz Gebreyes, really a One Health activity, big activity. It's really, really great activity looking at human health, animal health, environment, others. So what is One Health? And many of you, when I first heard saúde única saúde, not sistema, but it's like One Health, right? So what is saúde única? That we live in this interconnected, interdependent planet. Every year, more than 370,000 babies are born on this planet. United, the planet size is the same, babies get added, land use pattern. I will speak slowly, I hope, I'm sorry, I wish it was in Portuguese. The land use pattern, the deforestation, the climate change, the relationship between animals and humans every day is getting closer and closer, becoming more urban, more deforestation, more insect-borne diseases. Now you hear chikungunya here, right? Dengue, I knew in Brazil long time ago. Now we hear dengue in Florida, in Ethiopia, never been dengue before, and more temperate areas. So that interconnectedness is One Health. This is the United Nations definition of One Health. But One Health is beyond zoonotic diseases and not just human animal. The plant health, environmental health, a number of things associated to that. Just if I zoom into this, most of the One Health work, lots of social determinants work in Brazil, for example, or zoonotic disease, foodborne diseases, waterborne, vector-borne. Some of it we heard even this week are what you see here in terms of, prior to an epidemic happens, we're trying to prevent, right? Lots of discussion on prevention, on surveillance. Yesterday, my colleague Art was discussing many reasons why we do surveillance, and one of them is to prevent, right? So prevention, there are a number of activities we do. So that within the One Health, I'll show you another slide, but also looking at disease amplification within the animal health, working on that would help us to prevent from spillover into humans, right? So there are many things, many things discussed are toward that. And yesterday, Fiocruz colleague showed 60%, more than 60% of emerging infectious disease in humans are of animal origin. By the time you add vector and environment-related direct to human health, others is 75%, large and more and more and more. So this is a very important area. But with the global One Health, it's not just prevention. I would call preparedness. Preparedness goes throughout. between prevention, preparedness, and response. Response after outbreak occurred, outbreak response we're discussing, and I'll show one example from the United States today, is really we are responding. There is a One Health component on response. Many things we do within our global One Health in East Africa, and in Brazil, and in Thailand, Mexico, and Asia side, Thailand and other countries, is really capacity building, laboratory diagnostic capacity, surveillance capacity, clinical trial capacity. We have a fairly big project in Ethiopia we completed. So One Health is not just the traditional upstream, but also the downstream components. So here is a classical World Health Organization, but very important message here. Diseases often amplify in animals early on for a long period of time before they cause a human outbreak. This is the red one is on the human outbreak. So how can we be proactive? That's why Fiocruz and a number of partners are working on a number of epidemiologic investigations, molecular or non-molecular, working on these investigations, climate, environment, animals, to prevent. So if we really focus on this area, we will minimize when epidemics occur. Any large examples, right now, we're facing H5N1 in the United States, and globally we face Ebola or SARS, SARS-CoV-2, COVID, you name it, a long list of pathogens. So One Health focuses on that, but not only that, on the response side as well with global One Health. A couple of examples looking at the first one. This is antimicrobial resistance, the global trend. It's a major crisis now. I know a number of work happening. We were discussing yesterday with Janice here, but a number of work happening in Brazil in general. We collaborate a bit in Sao Paulo and UNIFESP and Anagales and many others. But looking at nosocomial community, a big problem. And this is a great study done about 7-8 years ago. By 2050, 10 million human deaths every year, each year, surpassing cancer and others due to antimicrobial resistance. It's a big crisis, right? So this is something that One Health plays a big role, because antibiotics are used in humans, in animals, but also in plants. In plants, often labeled as pesticides, but fruits, there are many oxytetracycline aminoglycosides used in fruits as well. In Florida or in the US, you come lots of antibiotic use in plants as well. And then the lots of aspects within the hospital use, the community use of antibiotics, and then the interaction of antibiotic resistance with, for example, disinfectants. We have studied a lot, Acinetobacter, mentioned recently the other day, Pseudomonas, becoming highly multi-drug resistance because of selective pressure on disinfectants in hospitals. We've done a lot of that work. That's One Health in hospitals. You don't need to have animal, you don't need to be a veterinarian to do One Health. It's a very important work we did in the hospital at Ohio State Wexner Medical. So we published a lot on this efflux pump, on molecular epidemiology of efflux pump. What you see on the right-hand side is National Academies published several years ago, the well-traveled salad. The salad, again, you don't have to fly anywhere to get a global issues, infectious disease and others. Chemical hazards sometimes, but biological hazards mainly. A bowl of salad on average in the United States could potentially originate from 36 countries. I think when I counted the countries on this map, could be 36 countries could originate. If you look at, for example, I never knew that we import croutons, this dry bread, from Brazil. You send the bread to us, dry bread. I didn't know that, but yeah, so it's again the interconnectedness of the planet, right? Just a bowl of salad can have ingredients from many, many countries. Of course, Brazil exports many food products to US. Poultry or beef, large product, or soybean, and many other things. Since I work on food safety, I know. Chapeco, how many of you been to Chapeco or Santa Catarina? Lots of chicken, and exported all over the world. And those chicken may carry salmonella, possible, right? So we work, and I will focus on specific demonstration of molecular epidemiology, but we at Ohio State University, the Global One Health Initiative, I and my colleagues founded and lead with partners, again, in many countries. We have this integrated approach as a university-based So, as a university-based initiative, we focus on training, learning students. Many students are here. It's wonderful, by the way. I'm very happy to see this connection of Fiocruz and Federal University of Bahia, and having this capacity building starts with the training. Shorter term, summer institutes, or longer term, PhD, postdocs, and many, by the way, many students and postdoctorate we hosted from Brazil, many states. Paraíba most, but many states. And then the other aspect is the research, the investigation, discovery, applied research. So our portfolio in many One Health areas, foodborne, waterborne, we work on cholera in Kenya, or many foodborne zoonotic, rabies, or many other diseases, or vector-borne leishmaniasis, including in Brazil, but our colleague have many countries in the world at leishmaniasis project. And many others, or non-communicable disease as well. Oncoviruses, like cervical cancer and HPV. There was a great talk we heard the other day. Or Epstein-Barr virus and non-Hodgkin's lymphoma. We work on different parts of the world, building capacity, building vaccine capacity, understanding, learning from each other, molecular diagnostics, genomics, epidemiology, or clinical trial, many other areas. So those are important applied research implementation priority, and coupled with outreach and engagement capacity or extension capacity. This one of the congress we led over the years is ICOFA Congress with Lee Riley himself. In Chiang Mai, we did workshop on molecular epidemiology. He was also a speaker. Chiang Mai we did Porto de Galinas, we did in Africa as well one time. So different, because we believe our principle is if we do these three core things and some supportive things like sandwich PhD, right? Many Brazilians come, or visiting scholarship, or other resources, stewardship. We build the workforce, healthcare workforce capacity. We build laboratory, hospital, field capacity, surveillance capacity, others. And then we share knowledge. That's when we fulfill our mission of building capable professionals to save this world, to make this world a better place for everyone. So all the young students this week asking us, wonderful. So we're looking for you as the future of this planet solving healthcare problems. So don't forget, use the One Health approach as you work with yours, but it's not just zoonotic, but whether it is what I showed you upstream or downstream areas. Okay, so far to the Ben? Okay. Any questions so far? Feel free to ask, interrupt. I'm okay. Just raise, ask me. I may, if I understand or respond, I'll get translated. Okay? So as we continue to the main part of my presentation, in molecular epidemiology, we said, vigilancia, of course, pesquisa, surto, outbreak purpose. These are the three. All of this we discussed in the last few days. So today I want to give examples of this: surveillance, vigilancia, pesquisa, or surto, using some of the work we did before, just to trigger our discussion and share some knowledge, but also please feel free to ask. So I'll use some Brazil and other examples. Just to start with, on surveillance part, for example, a key part of global surveillance network we have been working on. Brazil is a member, in Bahia, Fiocruz, others, COFPA, UFBA to be a member is this what called genome tracker. This is our network within the global one's arrangement with the US FDA. FDA is similar to Anvisa, right? More or less, more like Anvisa. But the FDA Center for Food Safety Applied Nutrition, the focus is FDA does lots of surveillance in the United States. But that's not enough, right? Because salmonella can come from different parts of the world, Campylobacter or other organisms. Therefore, they partner with us. Our partnership, it's a win-win partnership. We get isolates from different parts of the world. For example, this is a bit old. It's more now from Brazil, more than 100 isolates, or Ethiopia and Kenya. This is one example I want to give you. We have a bioproject site for our partnership on NCBI. You use NCBI for PubMed, right? Just like that. Instead of PubMed, you can use bioproject. So you will see our consortium's name, and for free, no dinero paid, for free, FDA does all the genomics. We've got more than 1,000 several years ago. We lost momentum a bit because of COVID, but that gets uploaded. We have the metadata. Then you can do lots of molecular epidemiology for surveillance. Sometimes you can detect outbreak after sequencing happened, or for research purpose, of course, many research. So I'll share with you one research. This is one example from raw milk in Brazil that, Daniel Monte was one of our joint PhD students, a number of Brazilian students under Professor Celso Oliveira, my colleague, long-time collaborator at Federal University of Paraíba, did. This is one of the many work, but I will show you another work which is very interesting and triggers some questions there. But many products come out of this, not just for student learning, but doing impact on the ground. When we did, in fact, milk work, it reminds me, I don't have slides now, but you all know FOMI Zero program, right? Lula da Silva 1.0, when several years ago, No Hunger project. When we worked in Paraíba, goat milk is more common northeast, right? Goat milk, the government gives muito dinero than cow milk. So some farmers mix goat milk with cow milk to sell, to get more price and sell as a goat milk. So government knew that happened, but wanted a proof. How do we solve that? So we needed a molecular diagnostic. So Professor Celso, my colleague, we discussed, and there was a way to develop a PCR method that detects if the milk has both bovine and caprine DNA. You get two bands. I don't have the slides, but very easy, but very good thing. We developed the PCR primers for each of the cow milk, goat milk. If the milk comes, you do a PCR. If you see two bands, that means there is adulteration of the milk. If you get one band, it might be goat milk, or if it's another band size, it's a cow milk, right? So very applicable, cost-effective, very direct benefit to the community. So there are many projects you can do like that. What I want to share with you today is this study. This study it interests me for many reasons. One reason is Salmonella enterica serovar Kentucky. We got, even in the US, multi-drug resistance in poultry circulating for a number of years. And then when we did East Africa and Brazil, others from East Africa, we get again multi-drug resistant ones. Janice mentioned multi-locus sequence typing, ST type. You see ST sequence type 198, right? So you think, this we thought, go back. Sequence type 198, maybe we were thinking this global clone spreading all over the world. Is it? This is one thing. In molecular epidemiology, the tools to address epidemiology are great, but you have to be careful which tool you use for what bacteria. And Albert was also mentioning to some extent. Not every genotyping method can be used for others. Initially thought, oh, this looks like a global clone. Is it or not? So that was part of the question. It's not necessarily molecular epidemiologic study, as Albert will discuss again, genomic evolutionary analysis, but more detailed molecular epidemiology follows up. But this genomic evolutionary analysis on this multi-drug resistant, which looks like a global clonal spread, but we are asking in this case. But the interesting part is the MDR ST198 in our project we get from East Africa, but the very capable people, look at them here is Mauro Saravia and Celso and many people are Alessandro, all that are from Brazil. East Africa isolates, but analyzed by Brazilian, very capable PhD postdocs that we have, and people from also Ethiopia, from Kenya, others. So I'll quickly show you the findings. So look at this. These are the isolates from Ethiopia, for example. We get this highly multi-drug resistance, including many of them have ciprofloxacin resistance, right? Fluoroquinolone is dangerous, right? So, and the similar, some multi-drug resistant ones in the US and other parts. So this is from our isolates done by Food and Drug Administration and CBI. Then we compared with the global other reported countries led by, that's Mauro, our student. So this is too much detail, but you look at now the power of this whole genome sequencing. In Paraiba, when they analyzed our students, you look at different plasmids. Like this morning, even Ron told us about the linear bacterial genome, the chromosomes, but also that extra chromosomal DNAs, different types of plasmid, Salmonella pathogenicity island, antimicrobial resistance genes. These are for aminoglycosides, sulfur, and all that. So you can look at this for all the comparative for our own project isolates and many other from NCBI. You can download and compare to do genomic analysis. We learned a lot on that, but quickly I want to ask you. Remember, we saw all this fluoroquinolone resistance, but when others there is no gene, what happened? We didn't find any gene. How did they become ciprofloxacin resistance? Anybody doing AMR work? You get acquired genes. What is the other mechanism for resistance? The word was mentioned even today. Because ciprofloxacin resistance a lot with mutation, changes in the base pair on the gyre A mutation of the topoisomerase in the genome. So students, you learn all of this. Sorry, it will be too much, maybe. But we were accounting for many of the resistances in one shot. The tool, right? To appreciate the tool. I'm appreciating of the tool. The genomic sequence done for each strain with a correct analysis by our own partners, Africa isolates being in Paraiba analyzed, sequence done in Maryland, United States. It's beautiful. I like this, the connectivity, and then analyzing and understanding the profiles of the genetic profiles of these isolates. Our project, but also compare these 248 isolates were compared. Okay. Then the big question, is this a global clone circulating in the world? ST198 is just one clone from poultry in Georgia or Atlanta or Connecticut or California, jumping to Ethiopia, Nigeria, Kenya, Brazil or others, or not? It was not. So using MLST, multi-local sequence typing, genotyping, for the purpose of Salmonella, Enterica, Serovar, Kentucky is dangerous, not a good idea, unless you do population genetic structure for epidemiologic scenario. Why? Janice mentioned it the other. The housekeeping genes are too conserved. Therefore, they look similar. They are not clonal. What this shows you from Celso Oliveira's lab, our collaborators, Mauro is a student, is really it shows several clades. The America clade here, look at this, USA and Brazil poultry chicken. USA and Mexico bovine is a clade. Another cow, bovine salmonella from USA, a different clade, though. Poultry USA, another clade. This is so different. And then there are Africa clades on the other side. It's more geographic because this is minimum spanning tree, phylogenetic analysis, Albert was speaking. Look at the different clades. Poultry, Nigeria, swine, Kenya, poultry, Nigeria. It's more of geography, but many clades. ST198 is not just one clone, several clones. And then there are what I call sporadic clones from pigs in Ethiopia that were identified. So we're able to conclude, as you can see here, I try to translate, Google Translate. So if it is not Brazilian Portuguese, excuse me. But there is no unique clone circulating globally. And we learned that. We can learn more detailed epidemiology. Just genomic analysis gives you that power to identify whether Kentucky ST198 is but multi-clones based on geography. Distribution is more geography, but per geography, maybe, yeah. If you look at poultry, USA, Brazil, as I say, Chapeco from Santa Catarina, chicken that went to, we have carried or vice versa, right? From where? So, yeah. Okay. So when we look at, okay, so I will stop there for the salmonella. Any question on the genomic evolutionary multi-drug resistant salmonella, please? Yeah, Albert. Right.
Wondwossen, just a quick question. Going back to the phenotypic ciprofloxacin resistance, please teach us what was the mechanism, given that there were not identifiable mutations in the gyrates gene?
Yeah. Thank you so much. So the different mechanisms of antimicrobial resistance. So there are some acquired genes like beta-lactamase for ampicillin. Other categories are acquired genes that break up beta-lactam classes, right? Amoxicillin, ampicillin, penicillin, others. The second group is modifying, target modification enzymes. Those are mostly for streptomycin and azithromycin, macrolides, and aminoglycosides, modifying enzymes. The third category is when ciprofloxacin you take, it goes to impact the bacteria, it attaches to the gyre, which supercoiling gene, right? So it attaches to the gyre, or so-called topoisomerase. Bacteria are smart. What do? Before the cipro comes in, some of the bacteria already switched. They modified their gyre. Certain location of the gene is mutated from adenine to guanine or something. It doesn't impact their survival, but it makes the ciprofloxacin not to be attached. So the mutation, okay? It's clear? Thank you, Albert. So.
And the additional one is flux enzymes, MDRs.
Efflux pump. Oh, thank you. The fourth one, we worked a lot, by the way. Multi-drug efflux pump. We were discussing with Janice about, like, when you have, during the COVID outbreak, for the whole world, all of us got impacted. On the respirators used in hospitals, others. Acinetobacter baumannii, Pseudomonas, they survive on that, right? So what we also found is a big impact. There is many publications on multi-drug efflux pump. It could be one gene, but efflux pumps, they come in, target antibiotics comes in, bacteria pumps it out quickly. Efflux, it takes it out. So it's a very important mechanism. We worked a lot on Acinetobacter on that in our hospital. So the four mechanisms, yes, yeah. Any other quick comment, question, anything? Okay. So it's more or less what I showed you so far is surveillance, again, with our global network. So let's see outbreak kind of example. So molecular epidemiology of this specific hypervirulent Clostridium difficile in the United States. It was US specific. There are some countries tried, but it is even in the US we get lots of diversity. This particular strain is called North American pulsed field type 1, pulsed field gel electrophoresis. Albert was showing yesterday pulsinate, you remember all that? So based on pulsinate, identified that. The same strain, if you do another method, ribotyping O27 on RAPD, many groups in Brazil do also rapid amplified polymorphic DNA, RAPD, or others. And then you can also zoom in, if it is toxigenic Clostridium, you can do toxinotyping using RFLP, or PCR of the toxin genes, PCR amplification, and then restriction fragment lingus polymorphism. You remember for tuberculosis, again, Janice will show you example. Lee Riley likes that. On his textbook, I remember vividly that RFLP for tuberculosis, for example, is a gold standard. You restriction digest the DNA of the TB, and then you use insertion sequence 6110. It's a repeated element present in the genome. And then depending on the profile of the band, Janice showed it. Again, Janice, are you in the room, by the way? But I was using her lecture as a background information. Then that binding pattern, you can say, is it a clonal? Is it a BCG? Is it B37? Is this epidemic to TB, or is it endemic one passing? Similar to that, there is a PCR RFLP, restriction fragment lingus polymorphism type, that you do toxinotype. I will mention later on, maybe to Mittermeier, Alberto, Luciano is. By the way, we have a number of lecture videos on molecular epidemiology, including Lee Riley, so mine, many. We can share with you, maybe students too, just the detailed reference. But you can use this. But for now, my focus is what happens to this outbreak, and how did we approach it? I never planned to work on anaerobic organism. I am a salmonella guy, foodborne, zoonotic, never taught anaerobic, but there was this outbreak. So CDC asked us to collaborate, bringing different scientists working in different areas because of this community-acquired unique strain. So quickly learned about how Clostridium difficile works, how Clostridium difficile associated diarrhea pathogenesis. The host, you remember day one, again, Albert, for students, you are here. The pathogen, we have to understand. The environment, we have to understand. Understanding that would help me doing effective molecular epidemiology about this outbreak. What happens to the host? These spores of anaerobic organism swallowed, but typically the classical endemic Clostridium difficile, if the patient takes proton pump inhibitor in hospital, typically, if there is a bile, right? So if it's proton pump inhibitor, the gastric pH goes up, right? Alkalinity, right? Alkalinized, then the spore passes. When it meets bile, it gets to vegetative form. Once it goes, then damage to intestine somehow because of different reasons, ulcerative colitis. This is for the physicians, but for epidemiologists, I don't need to know. I need to know some part of this. Okay, I know that. What happens on the pathogen, the bacteria itself? I am epidemiologist, I don't need to know the detail, but from the microbiologists already published information, what did we learn? There is this pathogenicity locus, 19.6 kilobase pair chromosomal region. The blue one, TCD8, TCDB, are the key toxins, enterotoxin and cytotoxin. Remember, this outbreak is now hypervirulent, so we need to understand what's happening. And then there are positive and negative regulators. TCDC, and that's down regulator. TCDD, up regulator of the toxin. If TCDD is expressed high, lots of toxin is released. If TCDC is high, the toxin level goes down. It's a balancing act. But what if there is a mutation on the TCDC or in one of them? Then either it goes, virulence goes down or it goes up. Okay, so far? Then what is happening in the gut ecosystem, in the environment? Disruption of intestinal microflora, if there is by what? Typically oral antibiotic. If we're taking oral antibiotic for something, oh, I have a flu, I went to my primary care physician, had some respite. At one point, I remember urgent care I went and I got ciprofloxacin. I said, why is this? The nurse said, I want to talk to the physician. What made him decide on this viral looking respirator to give me cipro? But anyway, that is a different discussion. But the disruption of intestine by antimicrobial, if these things are fulfilled, you get C. difficile, Clostridium difficile associated diarrhea. So what happened in the United States around 2007 is historically old age. Yannick were discussing about elderly people, elderly hospitalized long term, taking lots of antibiotics or proton pump inhibitors. They get Clostridium difficile associated diarrhea, but not anymore. Or not, they get it still, but in this case, community acquired infections came with that hypervirulent strain, pulsed field type 1. It has 18 base pair deletion in the downregulator. The downregulator has mutation. That means it's not functioning well. Lots of enterotoxin, cytotoxin produced. Again, this is too much, a little bit information from molecular epidemiology standpoint, but it helps us. So by 2008, lots of states were reporting it. Everybody was scrambling. Where is this coming? From pigs? Wondwossen Gebreyes works a lot on pigs. North Carolina, Ohio, Wisconsin. We did lots of it. From horses or from different people, CDC brought us together and did work. So quickly I'll show you. So we did more of a serial cross-section study design, if Arthur agrees. If that's a good study design, but talk to analytic epidemiologists, and then did, for example, we looked at the different toxinogens. Yes, the pig strains we get isolates. They have both toxins. Some of them are one of toxin missing or non-toxigenic, some, but they have that. They have binary toxin. We don't know the function. They have a deletion in the downregulator, but a different, not 18 base pair. 39 base pair. We don't know, we're investigating, it's epidemiology. And then we looked at from human isolates we got in our project were 24 isolates. From pigs we got more, but at this stage 84. We looked at what is causing macrolide and tetracycline resistance. Some similarity on macrolide resistance, some similarity on tetracycline, but like TET-W was very common in pigs, but we didn't find from any humans. Again, this is exploratory phase. Okay, then we got deeper, doing PCR RFLP. This is a classical genotyping method. Rupnik et al. Somebody published and everybody uses. So we did these 4 restriction enzymes amplifying as the toxin genes and then doing restriction fragment linked polymorphism. What did we get? Remember that outbreak strain causing community acquired infection is toxinotype 3. The pattern is toxinotype 3. Did we find any toxinotype 3? No. And then we are already having a clue that, based on the sample size, all that calculation, all the basic epidemiologic principle respected, by the way. Doing just molecular epidemiology doesn't entitle us to do, right? We have to follow the basic epidemiologic principle. But what we got was a bit different, but we got some interesting toxinotypes with 39 base pair deletion mutation, but it doesn't look like the outbreak strains that we are getting. The same thing with others. It was very difficult one. And then pulsed field gel electrophoresis. Others showed it, we showed it in pigs and humans. There are some similar ones, but their toxinotype 5 is the predominant we're seeing. Not the outbreak strain. So we already know now by then, it's not really the pigs causing in those communities that, and then the deletion. So just again, very shortly to show you, this is an outbreak scenario, but in our case, it does not look like outbreak is caused because we didn't find any North American pulsed field type 1. So you see that using these tools collectively, amplification, from unknown to known, right? Gene present, absent. Looking at PCR RFLP, you look at the different profiles. Already we know it's Clostridium difficile, but comparing it, is it the outbreak type or not, which is not in our case. Looking at further with PFGE further solidifies that finding, because all of these tools have better discriminatory power. Imagine if we were just isolating Clostridium difficile and looking at odds ratio and many, which we did, by the way. When it comes to piglets and humans, there is a high correlation, very high association. The odds of finding Clostridium difficile in farmers which raise small pigs, the piglets, much higher. And you think, oh, this is, but the moment you zoom in high resolution methods to get deeper in the bacterial strain, it's different strain. Okay, so far? Okay, I'm going to transition to one more research one. Now, we saw genomic surveillance in Salmonella, more of surveillance. We saw now outbreak and how we used molecular tools, and we'll transition to research. But in between, any Clostridium question or anything from Salmonella, Clostridium? Okay, all right. So let's move on now. More of molecular application. Again, another quick example in the interest of time, using what we initially called Campylobacter genomics and environmental enteric dysfunction. Environmental enteric dysfunction causes stunting. What's in Portuguese? Como se chama? Yeah. Children, not constipation. Children not growing as expected. Yeah. So stunted. Okay. Okay. So I think I have later on Google Translate. We'll find the word. So this project was led by University of Florida as nanism. Okay, you got it. Nanism. Okay. So University of Florida were collaborating with Professor Ari Havelar. There are also anthropometrics. There are lots of interest from their side. And there is lots of stunting in different parts of the world. So some reports previously showed Campylobacter, another important foodborne bacteria, very common in chicken and pigs, others, and then common in humans is important, right? So that was hypothesized, that was published as a very important agent for nanism. Okay, so we did a series of publications, and the study in Ethiopia was because there was a specific region with a very high rate of stunting. So 25% of children globally get stunted. 35% in Africa. Ethiopia case was even worse than the average in Africa. 38% in Ethiopia. So in the area, many things, the impact is so big. So quickly, I'll show you just the findings. But again, we launched a longitudinal study, a cohort, prospective, following 10 communities. There is a more of control community, but 10 communities within the regions that's heavily impacted. We looked at infants. The target is little children, 11 to 13 months of infants. This was the initial, but we got much more larger sample size. But we looked at Campylobacter positivity with enrichment, others. If you look at on this table, is a little bit more informative. There was a group from Florida, again, that's doing the LAZ score, which is about the stunting score. Which children were severely stunted, LAZ score -3 or below, or less severely. When we look at Campylobacter, there is not much correlation. Campylobacter is common, whether it is severely stunted or mild stunted. Okay? So far, okay? So it did failed, children, chicken, and but also other animals.
Nanismo nutricional
Okay.
is stunting.
All right, okay. All right. Thank you, okay. So there was we didn't find, based on the observational study, we didn't find many things we did, by the way. But today I'll just show you a few slides. doesn't look like just Campylobacter highly correlated to stunting or no. The odds ratio was crossing, all very large standard deviation. There was no statistical association, all that. Okay, so we went ahead and did a further, more detailed study. Again, looking at the infants were our target, but looking at their household, maybe child, mother, caregiver, mothers, and animals, livestock in this targeted cohorts, the cohorts following, including the animals, humans, we follow them regularly, sampling, and then once we get positive, about 280 metagenomics. Why did we need to do metagenomics? Why don't we do just like genomics like Salmonella? What do you think? It's been mentioned too. Anybody who can answer why in the room or in online? Why do we need to do metagenomics? What's the difference between genomics and metagenomics? What do you think the reason?
Giovanni.
No. Exactly. All genetic material in the
Sequence
gut
the whole
ecosystem.
genomic material.
Yeah.
Okay.
Whole genomic material in the gut ecosystem, we can find much more information. If it's genomics only about Campylobacter, it's important, but we already know from observational study, but we still did Campylobacter in depth. But this gives you in-depth Campylobacter because there are many species. If you do culture, you cannot pick all species, but also muito organismo. Okay, so it could be bacteria, virus, parasiticos, many others, protozoa, others. Okay, so we did. That's why we did. So quickly again, some of these molecular things are too detailed information, but just quickly to show you. Even if you can't read it, you trust me. There are many organisms. These are children. Each column is children. Heat map. What organisms did we find? Many organisms. Campylobacter is among the 25 top at the genus level, but there are many other organisms. Look at here. The infants on the right-hand side, what you see, the blue one, infants had Shigella, Salmonella, Vibrio cholerae, Giardia, Amoeba. Okay, co-infection. In many cases, co-infection. In some cases, it could be one or two organisms. And then other family members and cattle had many others. Vibrio melliticus, other amoeba, Cryptosporidium. We found Cryptosporidium parvum in some of the children as well, but this one doesn't show. But based on the abundance, the metagenomes, these were the predominant, the main ones. There are longer list of organisms, okay? So we looked at further Campylobacter. If you look at the species level, Campylobacter, many species of Campylo in one child could be impacted by many. You see these different colors? Each of these are children, infant, children. Some of the infants were impacted by more than 3 or 4 or 5 Campylobacter. Some of them with 2, Campylobacter intestinalis and a newer strain RM we found, or a potential species. Okay? All of these data collected and published, and there is new one just got accepted by my former postdoc, showed us that it's multifactorial. Environmental enteric dysfunction is not easy to. Stunting, it's caused. That dysfunction causes the intestinal lining less absorption, and then lots of diarrhea, and the nutrients they consume is not absorbed very well. There is a dysfunction, but the cause is not one bacteria or one virus. We believe it's multifactorial. So this is a pure research, but that would lead to many things in the future, we believe. So there are many other findings. Okay? Yes.
I just have a question. Were they consuming the goats, the chickens around the area there?
Thank you so much. Were they consuming the goats, the chicken in the area? Very good question, because it reminds me to answer. That's why I like questions. Is when people ask, I remember some important things I forgot, right? So, number one, yes, food consumption. Oh yeah, goats, chicken, others consume. Children, even as I showed one photo earlier, they don't have toys or iPhone most likely in the rural areas. The chicken they play with the chicken. There is a direct transmission. They play with chicken, and children are very common. Another part is the hygienic situation. In that part of Ethiopia, when I grew, there was a big lake. People go, wash, take a bath, big lake, Haramaya Lake. There is a university there. Right now, that lake, when I went 5 years ago for this project, you can see the lake only on Google Map. You see blue, because there is nothing, no water on the ground. So that impacts water, sanitation, hygiene. So yeah, so it's very important. Yes, consumption. There is interaction with animals. On the first slide on this portion I showed, animals cohabitate in the same room with families. In the same room. You can see chicken, cows in the same room. They sleep. It's common. In my country, Ethiopia, that's way of life in many rural parts. So yeah, there's multiple ways of human-animal interaction. That's why this is very classical One Health project, dealing with environment, dealing with animals, but the ultimate goal is the public health part. Great question. Any other? Okay. So yeah, it's complex. So I will almost wrap up, and I'll be happy to.
I think we still have another 5 minutes or so.
5 minutes, perfect. So I'll be wrapping up. So thank you so much. So far what I showed you, remember that upstream, One Health, a lot what I showed you is here. But there is a lot we can do on this part. Yesterday again, I think Arthur, before yesterday was mentioning IHR, International Health Regulation, what's reported to WHO. Global One Health is that transdisciplinary connection. We do a number of projects on that, advancing laboratory, surveillance, even emergency management to meet IHR requirements. Number of countries fulfilling, but a lot of capacity building needs. That would help us all, prevention, preparedness, and response for future. We work on infection prevention control. We work on a number of other areas, on global antibiotic resistance network, which I am advocating Brazil be part of that network, is CDC. It's a great project, by the way. We're connecting Chile is part of it, Ethiopia, India, many others. So to finalize, this is almost my functional slide, is really using molecular epidemiology in global One Health. Why don't we just stay analytic observational study? Often because you need that discriminatory power. The power of molecular tools in epidemiology to discriminate infectious diseases that you cannot get from phenotypic investigation. Culture, antibiogram limited, right? Genotyping gives you more power. Genomics now even revolutionized it, but other. High resolution, you see, we zoomed into the PALOC region of Clostridium difficile, for example, right? You can have high resolution rather than culture others, so that's very important. High throughputness is very important, especially if you are working with a surveillance area, large sample. How many samples can you do genotyping in 24 hours? You may have, right? So if you work for Governor do Brazil or do Bahia, and you have a large surveillance plan, high throughput. Many methods give you high throughput, not all methods. A speed, some of them give you rapid, right? RT-PCR or something like that, rather than 2 days, 3 days culture, especially for outbreak scenarios. And sometimes the value for money, the cost benefit, costo beneficio, right? So if you do metagenomics, imagine there is no in this world that have enough time to do all the cryptosporidium, all the Shigella, but it's cost effective. And it allows us to do a more conclusive, more solidly conclusive, resultados mais conclusivos. So, but when you select the abordages, oh, that's difficult. Moleculares depends on the epidemiology, depend on scenario epidemiological. You have to be careful. Just because Professor Smith from United Kingdom published, you can't use that same method, two different methods, right? So, okay. With that, I will stop again. Lots of great sponsors. I want to remind, like some of them, foundations in Brazil, CNPq, CAPS, many other, Governor of Brazil, ICGEB funded us for here in Brazil, UNU Biolac, Biotechnology for Latin America, many others, INSA, Semiaridos, Institute Semiaridos, many.
Now you can include SGSA.
Oh, yeah, yes.
Okay.
We can add, absolutely. We'll be happy. Prazer. Yeah, so we have been doing molecular epidemiology course. 2009 is the course. 2007 we start project. 2009, every other year we were doing. This is the first batch in Federal University of Paraíba, but it grew later on. 2011, American Society of Microbiology funded. 2013, we had a global people from Africa, Asia, others in Porto de Galinhas. That's when I knew Lee 2004, but he came from here to join us. But and then 2017, we did FDA Genome Tracker course in João Pessoa. So muito obrigado. So thank you so much.
Thank you very much, Dr. Gebreyes, for a really wonderful presentation, and I think very important for not only the course but for Instituto Salomões because of the, introducing, the One Health hasn't been perhaps a major emphasis here, and it's time that many of us should work on that. So I think we have time for a couple questions. Is there a break after this? Okay, so maybe time for some questions from the students.
Hello. I'm here. Thank you, Professor, for the lecture. And I have a curiosity. I'm not inscribed in the course for this year. I did it a few years ago. And for the Clostridium difficile outbreak, was there ever a resolution to discover what was the origin of the pathogen? I know that for most of the situations, we don't discover where it started, but maybe for this situation, there was an answer.
Yeah, thank you. Muito obrigado. Very important question. So was there a discovery of the C. difficile NAP1 outbreak? After 3-4 years, we met back in Atlanta at CDC. The resolution we reached there is all of the animal sources we hypothesized were not the sources. We believe that hypervirulence somewhere as this bacteria reproduce within the human intestine, they mutate. And that mutation, especially remember that 18 base pair mutation is dangerous. Why? It is a multiple of 3. 3 is amino acids, right? 6 amino acids are out. So it's significant frame shift mutation that's caused what we believe is it's just human, the evolution of the bacteria within the human population.
Another question?
Sorry, my English is not good, but I'm trying to put my question. So you study about the journey of residency, but you study also about the microRNA, not codified gene, the impact about this gene, microRNA.
Micro? Yeah. By the way, your English is better than my Portuguese, so you are better. But no, we have not looked at microRNA. I cannot comment much, really. Yeah, I don't know. Really. I feel that would be nice for future scientists, rising stars to investigate.
After those results, the crew, the team, they gave to the people in the area some recommendations about it to try to prevent those disease.
Oh, absolutely, yes. So to prevent future disease expansion, transmission, a number of measures. A number of measures at community level, at hospital level, because from the communities they come to the hospital, right? Yes, which is not really part of but my colleagues on the infectious disease unit, infection epidemiology, we collaborate. We even used our children hospital. We were seeing some hypervirulent Clostridium difficile, some toxigenic. But remember, even though like in the Clostridium difficile case, we focus on the outbreak strain, we still were getting potentially hypervirulent other strains. So there was education on that. Yes, a number of intervention. Still, yes. The sad part is some people even died because of this strain before we can take measures. Yeah.
So we have a rule at Yale that in these type of seminars, students first, professors last. Any other students have questions? No? Good.
That's me. So thank you so much for your presentation. I'd like to know if you find any relation between genotypes and multi-drug resistance in C. diff, because multi-drug resistance is another issue in this agent.
Yeah.
So I just wonder if you find anything.
Yeah, thank you, Yannick. Very good question. Again, that's one of my slides I didn't include for interest of time. Some of the strains we got were vancomycin resistant, dangerous, coupled with macrolide resistance and metronidazole resistance. Yes, metronidazole, not good. The metronidazole, so we found some, that's correct. But what happened is, even the toxinotype 5, the predominant we got, some of them are metronidazole vancomycin resistant, some were not. The tetracycline is okay, some of them are constitutively resistant, but yes, there was some association. But not all those strains are vanco or metronidazole resistant. Important issue, yeah.
Okay, so I just want, before thanking, I just want to make one comment, and this is for the students. So this was a very important talk underlying some of the uses of molecular epidemiology. And one which came out was certainly, risk attribution, or source attribution, particularly in this several examples that you had, whether it was a negative example of pigs, for C. difficile, right, or salmonella, yeah. and Campylobacter, well, animals for Campylobacter-mediated enteric enteropathy. And just the second thing is that, really the use of what Lee would call next generation molecular epidemiology, which is really whole genome, metagenomics and whole genome sequencing, especially teasing out. It's a good example where doing an analysis that couldn't have been done without metagenomics and next generation sequencing, because something as important as enteric enteropathy is multifactorial, and many of the agents may not be culturable, right, in the microbiome. So just to go back to those kind of core principles that Lee Riley had put in his books about the types of analysis you can do. So this is really very great examples. And this is not as important. It used to be very important in Brazil, in Ceará, in much of the Northeast, but this is the nanismo nutricional, or stunting. So remember that for the students, there's marasmus, there's kwashiorkor, which is protein, carbohydrate, and then protein malnutrition. But the most important form of malnutrition is stunting, right? And really kind of just really important studies that you had showed. And I guess these were funded by the Gates Foundation, right? On in making that link in that source attribution.
Exactly. Very good. Excellent point. Yeah.
Okay. So thank you very much.
Has a word here. I think very few knows about this word. It's a gabiru. That's someone that's not get food sufficient to grow.
Oh,
He's a Gabiru.
Gabiru, okay, Gabiru. Okay.
Joyce, he wants to say. Why don't you close that?
Only the last question about the kids and the metagenomic analysis. They found a lot of kids with Shigella. So it was like a subclinical infection because as a microbiologist, Shigella is not carried by humans. So
Yeah, no, not just subclinical. They were clinical too, because they have diarrhea, but we don't know, right? So the metagenomics helped us. Many times they have diarrhea, recover. Some maybe, just carriage of Shigella, but there are multiple. Shigella dysenteries, I had when I was a child myself. It was very bad, bloody stool, diarrhea. So some were clinical, but we have a detailed demographic and findings in that one. There are many, and I mentioned it a little bit with diarrhea. But some of it we don't know exactly if it's Shigella because we don't culture later on after that to really find if it was the Shigella causing the diarrhea. You get my point? So children number X may have Shigella, may have this, cryptosporidium, this, and then diarrhea. But which one is causing? Not necessarily we know because of the research. It doesn't follow up further to culture.
okay.
But very interesting cohort. It's 10 districts, many cohort. It's one cohort of 10 districts and multiple small units that were following. Very interesting even to do many other studies. Yeah, thank you.
Okay, thank you, Professor.
Thank you, Janice.
Como o professor... Encerra, então.
Então, gente, a gente encerra agora essa parte, né, da sessão científica. É muito importante para a gente ter a participação dos cursos e a participação desses eventos que enriquecem a sessão científica aqui do IGM. Então, obrigada pela participação do professor. Cadê ele? Thank you very much. E vamos continuar com o curso que realmente traz uma qualidade e uma relevância muito importante aqui para o IGM. Obrigada e agradeço também a organização.
Como o professor Wondwossen não estava na foto oficial, vamos ligeiro lá para frente tirar uma foto oficial com ele. Ok? We need to take the official photo with you too.